Okay, so what we're going to be doing today is we will be making a um, poster template that you will use to make your final poster for next week's presentation. Now, the idea behind next week's presentation is that you will be able to share your screen. So you will share your poster through your screen, and then you will be talking through your mic and presenting the poster, just like you would have if you were in the class, except virtually. So that means that everyone here should have a working mic. If you do not have a working mic, if you or your partner does not have a working mic, send me an email and I will give you an alternate for how you will be able to present. You will still be sharing your screen the same way, but I will give you allowance for not having a mic so you can present a different way and post that video separately as well for people to see. Now, for the poster, I have some instructions up, obviously, under the assignments tab, but we will go ahead and make the basic template together so you know how to get started on it. Um, okay, so if you want to look at the instructions along with me as I go through the making of the template, you can go ahead and open up the poster draft with the final figures. And so this is actually the same PowerPoint that we had just talked about. Don't open that one, sorry. My internet is having some issues apparently. Assignments. Go to poster. There we go. So you are going to be opening up the poster presentation that says that's a PowerPoint presentation, and there are some guidelines and instructions in a separate document as well. I'm going to go through the guidelines and instruction Word document first. That's the one that you can refer to back and forth for a quick check on making sure you're doing things correctly. So if once it opens up, it is obviously telling you that, originally we were going to print it out. So it gives you all those guidelines. Don't worry about those. Just go to the general tips for preparing your poster. So the big idea behind the poster should be that it is self-explanatory. You should not have to do a lot of talking to explain it. Anything that is needed to communicate the basic results and conclusions of your data should be readily available on that poster. Having said that, you want to make sure that your poster is not wordy. So each word is precious on your poster. So you want to first maybe make a little draft where you write down everything you want to say in there, then reread that stuff that you've written and write it in the most succinct way, most, least wordy way, so that it gives you just the important information for that. Now, for your figures as well as for your text, we want to make sure that it is large enough for people to see in a poster. When they are standing there in front of it, they should be able to see at least, from at least 2 feet away, should be able to read it and understand what is written there, what is shown there. Um, so there are some minimum size requirements that we will follow, and those are given in the PowerPoint that I will show you next. So we'll talk about that, what percentage of your poster will be pictures compared to text. We will talk about that shortly as well. Um, but in general, when we think about it, we want our our data to shine. So obviously, like at least 50 to 60% of your poster will be taken up by images of your graphs, your figures with their titles and captions and whatnot. The remaining areas will be what will be for your background information, for the methodology, for your conclusions, those type of things. So if you think about one of those, the trifold posters like we used to do in, high school or middle school with for science fairs and stuff, the central portion is the larger area. All of that would be your results, and maybe some of the, like a third of the one of the other sides would be results as well. And then the first side, that little sliver on one side, would be your introductions and your methodology, and then the last portion would be your conclusions. And that's what we will work on. We'll create kind of like a trifold template when we get started in a minute. So you do want to read these points about planning your poster and how to do it. Um, so you have a little bit of pointers on what it is that you should be using. For your data and images, you want to use the best quality images, and you want to copy paste them appropriately so that they don't lose the resolution. Many times when you enlarge them, they lose the resolution, they become hazy if they are not copied and pasted properly. So we want to make sure they're not blurry and we can read everything on there. Um, you want to choose, you can choose any colors you want, it's fine. But you want to make sure that the background versus the text is different enough that they are easy to read. So you don't want to use a sky blue with white text, right? It won't work. You want to make sure that there is enough contrast between the text color and the background color. Um, and then obviously your text sizes should be correct throughout, and we'll talk about what those should be. For your background and introduction, you need to have it in. So now you're not writing an actual introduction. What you're doing is, if you go back and think about the N2A paper introduction, we talked about having a 5-paragraph essay format. So each paragraph would have started with some type of a topic sentence and ended with some type of a conclusion or whatever. So think about those topic sentences. And that's what you're writing now in your introduction. You're writing just those 3 to 5 bullet points that you would have made paragraphs out of in your actual introduction. So your introduction is going to be in bullets, along with, you can use images. For example, if you're doing mulberry, you can use an image of mulberry plant or mulberry fruit, along with the chemical structure of the actual compound in mulberry that has the anti-cancer effect or that has UV protective effect. You can add schematics of any type that you want to talk about. For example, if you're doing UV radiation, you can have a schematic to show how it may be affecting the body and why you want to protect yourself. Anything like that. So you can use it from an online source. Just make sure to cite it, and it can be a super tiny font, but it needs to be there on the image or in the text. So that would be your introduction. Your methodology, you can write either in bullet points, or you can make kind of a flow diagram or a schematic format. I'll show you some examples of what you can do. Then for each one of your figures, you are required to have no more than 3 multi-panel figures, total figures. At least one of them has to be multi-panel. Majority of your figures will be multi-panel. For each figure, what would have been the title of your figure will go on top of the figure in as a heading. Then you will have your figure, and then you will have your caption. And your caption should include any important information that is needed to understand that figure. So that would be your figures. And then for summary and conclusion, yes, multi-panel means that you have multiple types of data that, when thought about together, that brought together, give you one common conclusion. So we will make an example multi-panel figure in a few minutes when I get started on the actual template. And then for your conclusion, you will do the same thing where you highlight your main conclusions in a bullet point summary, not big paragraphs, not extremely wordy. And that would be essentially the poster that you will be working on. Okay. To prepare the layout, we want to make sure it's the correct size. So we are still going to prepare it as if we were going to prepare it for class. All those things will stay true. And I will show you how we will do that. All the fonts, including any fonts in your axes for your figures, have to be at least a 0.14. So you want 14 point or above font size. And 14 point is like the smallest you want to do. That's usually going to be just your axis. Everything else is going to be larger than that. Many times for your headings, you use like 44 or above. For your title, you'll probably do 60 or above. So it's going to be, large font sizes because it's we always print it out one time on just a 8x11 paper and look at it to make sure there are no issues with the way the formatting is done, the way the colors show up, the way the words show up, all those things. So it's a good way to look at them. So we do like a mini poster that way. And then we usually, finalize after we make all the corrections and typos and everything. Then we get the actual thing printed. Now, for your presentation, if you were doing it in class, it would have been 3 to 5 minute oral presentation, and that's the same. We do not want to go above 5 minutes for our poster presentation. It is, either in the middle of the day or in the late afternoon, happening out there. And people will go obviously towards the food first. They'll get their plates of food and then they wander over to these posters and they start having discussions with people and talking to people that they're interested with, talking about their posters. And so it's more a conversation. It shouldn't be where you are sitting there or standing there giving a 20-minute, instructional video on your poster. It should be a very quick, short presentation. For your presentation, you want to make sure when you will present to me and the class, you want to make sure you highlight the important points. You give me your aim and hypothesis. You give me the main conclusions from your figures, pointing out important details in your data that I should be focusing on. And you give me your final conclusion. That's it. Everything else that we want to know, we will ask you as a question. If we want to know more about your methods, we will ask you. If we want to know about your statistics and exactly how they were done, we will ask you. What you are going to present, you are a salesperson for your poster. You're going to stand there and you will talk about your data and sell me your story in the best way possible in a very short, succinct presentation and lively presentation. Now, typically, when we do it in person anywhere, we want to be dressed you can for our course many times if we coincide with some kind of a like Halloween or whatever, I even let people dress up according to their design of their experiment. You are, and I will give you the same. You can make that as part of your presentation if you want, or not, but you should be presenting professionally. Obviously, we are just going to be presenting through our mic or our webcam and our posters. So it won't be as important to make sure that you have the whole 9 yards and nice get up, but you still want to present the way you would in front of a conference and be professional about it. Any questions on these basic things? Okay, so we will move on to the actual poster. I thought I downloaded that. Let me open it up. Okay, so I'm not going to go through through every single slide in here, but I will talk about some important stuff. If you look at this first slide, you are going to present together. So both of you are required to do some bit of talking in person talking. But you will divide your work according to whatever it is that you want to, however you want to. If only one person has mic available and there's an issue with that, we will come up with a different way, but both people have to be part of the presentation. The presentation itself is about 35%, I believe 35% of your total grade. So to get that, you have to present. You have to present. Slide, it gives you a lot of different posters from various places as examples of how people present posters or show posters. Some are extremely wordy, some are have more images on them, some are more creative, some have more logical flow. But you can look through some of these as examples to kind of think about how people present the posters. Posters do lend to get, it is a more creative way to present your data. The idea is that you are going to be showcasing your experiment and presenting your experiment in a way that we would want to be excited about it and we would want to know more about that. So you want to make it in lively colors and interesting um, fonts and interesting pictures so that people are pulled towards it. People come to you and gather around you and ask you about it and want to know more. Okay, somehow it is not changing. There we go. Okay, so when you present your poster, the idea again is not to present every single word that's listed there, because the poster is the visual point. That is your data that people are going to look at. It is to point out things that are of interest in there. So people are pulled towards it and visually go towards it and look at it more and discuss it more. And that's what you want to do with that. So we don't need to worry about that right now. One of the reasons that we do poster presentation is obviously it is part of the biology department's goals is to get you ready for future life, working life, and part of that is to enhance your public speaking skills. And this involves practicing for that as well. It also helps you learn how to organize your data, present your information in different ways other than just writing a paper about it. Now, again, when you are making your poster, the first thing you want to do is get your story straight. So you want to make a logical flow chart of sorts about your story, going from your hypothesis and aim, and then what data would you like to present first, second, third, and how that story flows once you present it that way. It should be visually appealing. Should be logical and easy to follow. And it should be, each figure should be a standalone so that people can look at it and get their conclusions out of it without having to read a bunch of stuff or learn a lot of new things. Here is an example of how you would do that. So you have your overall goal of some sort. In this case, it could be you are examining an anti-cancer activity of a new plant, its mechanism of action. Then there are many things we can do with it, right? So the three goals that I want to present in my three images, the first one is that I want to test the anti-activity of the cancer activity of the plant as a whole. So I want to present its methods and results according to that. Then I want to logically want to know exactly what part of my plant is having that activity, is causing that effect. So I identify the extracts, separate them, and then look at them again. So I have the methods and results for that. And then I have my last goal in which I have now extracted the compound, and isolated it, and now I want to know, well, how does it work exactly? What does it do? So then I study the mechanism of action in my third figure, and that's my method selection. Another way would be to have my overall goal followed by my methodology, where I have a flow chart and showing all three parts of it in one time. And then I switch it over to my results, and I divide my results like I've just shown you. We have figure one describing the anti-activity, cancer activity of the whole plant, figure two talking about individual extracts, figure three looking at the mechanism of action, and then I may have a figure four, which gives a schematic and brings it all together. So what you will have is the different sections here. You have the first thing that you'll have is your title of your abstract of your poster. You will also have the names of both your partners, and you will have the affiliations and university information. Department of Biology will show you how to do it. Then you must have an introduction area. Then you will separate out your hypothesis, goals, specific aim, whatever you want to call it, separately at the end of your introduction. You will have methodology, you will have your results, discussion, and future directions, and then references and acknowledgements if needed. References you will have. You can use a small font for those if needed. You don't have to adhere to the at least 14-point font for the references section. For your rubric, we are going to be judged on 6 areas. One is the actual PowerPoint of your poster. The second would be your introduction and the hypothesis, the way you presented that. The third would be the way you presented your methods, if they were correct, and if they were presented in a easy to follow way. The fourth would be your results, and then the final would be your conclusion and future work section. I will also grade you on your overall presentation and your handling of the questions that we ask you at the end of your presentation. We will have the 2 partner groups present back to back, and then we will ask questions together for those 2 groups. That's how it will work. For your poster appearance, want it to be more heavy on images and your actual figures than words. It is hard for people to go through 200 posters in a poster, meeting and go through and read all these words. They are going to be focusing on your images, your headings. Those need to be clear. And then minimum use of words otherwise. You have a maximum of 4 images, including the schematic that you will make at the end with the pathways. So that means 3 figures for your data and 1 for your schematic. Um, yes. For your appearance, you want to make sure again that you don't use a lot of words and paragraphs. Instead, use bullet points whenever you can. Your figures, you want to make sure that you mix and match them. Don't make everything a bar graph. Don't make everything a line graph. Combine them together. Think of creative ways to present your data so that it isn't all the same thing. Yes. And then obviously, if you want to make sure that your flow is easy to follow. So if you're going horizontally or vertically, we should be easily able to see what comes next. If you need, you can number some of the sections as figure 1, figure 2, figure 3 to help us, guide us the way you want us to look at them. Here are the minimum font sizes, some of those. So the title should be a very large font. The minimum size we say is 60, but usually you want to use 90 point for your font. The names underneath, yours and your partners are going to be smaller. So if you have your title at 90, the names would be at 72. If you have the title at 72, the names would be at 60. Yes. I saw some comments come up. I'll look at them in a second. The idea is that if you had this poster printed, we should be able to stand 4 or 5 feet away and still be able to read majority of the areas without going close to it. Okay, let me look at some of the chat that was coming up before I go forward. Yes, it is being recorded. I will be posting it afterwards. Okay. Okay, going back to this. So going back to each section specifically, and this is something important that you guys should look at and revise your titles. If you provided me with a title, many of you got that same comment. When you're writing the title, especially in a poster, also in the paper, you want to include both the independent as well as the dependent variable in your title. It should give us the final conclusion from your study. So your topic is effect of retinoic acid treatment on neuroblast cells. That is not a good title, however. Your title should be that retinoic acid induces differentiation of neuroblast into neurons or does not promote neural differentiation, something like that. It should give the final conclusion from your study. So be aware of that. Many of you, when you were writing your title, just wrote effect of mulberry on, MTA468 cells. That's not a title. A title should be taking into account the final conclusion that you saw and doesn't necessarily need to focus on MDA 468. Could be just talking about in breast carcinoma cells. And then in your methods, you can say that you were using the MTA 468 cell line. So the title is going to give your overall big conclusion. For your names and affiliations, you will have the names of both partners. You will have your usually we have the department, university name underneath. So in your case, it will say, Molecular and Cellular Biology Lab, Department of Biology, University of North Florida. And then many times we will have like an address for it. So it would say like Jacksonville, Florida. And then sometimes they require us to add email addresses. Obviously, you don't need that. Many times we have logos on that title area in the top of our posters. We will add those. We will add a UNF logo on one side, and then you will be able to add something else of your choice on the other side. For the actual text, you want to make sure your text remains consistent. So if your one heading is font size 48 or 60, then all your headings should be the same way. Don't make half your headings, 60 and half of them 20, right? So you want to make sure that they are comparable. Sometimes you will need to change your font sizes and your headings, for example, for your figure headings, because they are more wordy. In that case, make sure that all the figure headings are the same and then the side headings are the same so that there is some continuity and overall design looks good of your poster. You want to always spell check multiple times to make sure that you have no typos and no issues with your poster at the end of the day. For your introduction, you are writing, like I said, you are writing the introduction in words. And then your intro will end with a separate section that your objective, aim, goal, hypothesis, whatever you want to call it, is going to be that one sentence that you have been writing for your hypothesis. Or you can write a couple of sentences to basically talk about why did you do this and what did you expect. For your methods, you are going to, again, you can do one of two things. You can include a flow chart to describe your methods, to talk about the different set one, set two, right? and what was done in each set. You could do a bullet with the by technique. So you could have, your cell line, the treatments, and then have the various techniques. So you could have relative growth analysis, protein extraction, caspase activity, things like that. Make sure you tell what statistics were done in there as part of your experiment. Just summary take home bullet points on the side, and that's it. Nothing more than that. Make sure you tie it back into your original hypothesis and talk about whether it worked or did not work, and why or why not. Also, tie it to larger world problems by talking about how is this applicable? Why is this important? Maybe the internet is going a little crazy with the thunderstorm or whatever. It is recording. I hope it's recording without skipping. If not, I will be recording it again tomorrow as well. And I did two separate recordings today. I'm doing a Canvas Studio recording and I'm doing the Zoom recording. So I'll post both. Maybe one is clearer than the other. Okay? Okay, let's go. It is the due date originally was going to be today, obviously, because we were going to do it in class. It is flexible. All the due dates at this point, except for your actual sitting down and presenting the poster, is are flexible. I do ask that you turn in your draft at least 48 hours before your meeting, your actual presentation. So you have, well, actually 72 hours before your actual presentation. So you have time to make changes based on my corrections or my comments. You're going to be creating a PowerPoint poster, which is how we were going to make it anyway. Everything is going to be done the same way as we were going to do before, except you're not printing the poster. You will be sharing your screen just like I am, and you will be activating your mic, you and your partner, and you will present it together. Yes, you will be uploading the PowerPoint by before your class as well, but you will be sharing your screen to present it. Is that good? This is already uploaded. This is under assignments. You can find it for your final poster. It will be there. There are two documents. There's a Word document and a PowerPoint. You are making a PowerPoint poster. It's not a Word document. It's always a PowerPoint poster. Yeah. I'll show you in a second how you'll do it. Okay, so tied back to the real world problem. Again, in your summary, make sure you have your one or two main conclusions. Those are your first two bullet points. Then you have a bullet point talking about whether it worked with your hypothesis or not. If it defended your hypothesis or not, and then give a fourth or fifth point talking about how why care about this? What's the big world problem that it can solve? Or how can it help understand something or help our health, whatever it is. And then it would be helpful if you could if you would write a point or two about future work. What would you do next? If not, I would ask you. So be prepared to answer that question regardless. You can present it on the poster or you can just be prepared to answer it. Yeah, the internet also, if you look at it, at least the screen says internet unstable every now and then. I don't know why it's not coming down. Okay, so again, for slides, we don't really need to know too much about. Um, the references, if you have used a reference, you need to cite it. You can use it within the text the way we've done before, where you write the last name and the year, and then you can make a small section at the end where you give the references and their little information. You don't need to give them in large format. You don't need to write the whole article name and everything. Just write the name of the first couple of authors at all, write the year it was published, and write the DOI of the paper. That's all you need to minimize usage of space. Normally, when we do it, we acknowledge people that we want to thank. It could be mentors, lab mates, people that have helped you with technical work, done other stuff for you for the paper or for the poster. Now, we're going to be doing this. You can see that I have some instructions on how to do it in the PowerPoint as well. I'm not going to go through these because I'm going to be showing you how to do all of that. But just to show you a couple of things in here, which you can then look through, it gives you how you should make sure what type of formats are good for good resolution of pictures so they don't look pixelated. You want to make sure that you are always stretching them to maintain their proportions so they don't look messed up. So always enlarge proportionally. You want to make sure that your text boxes and your labeling are done appropriately. You can change their colors. You can do a lot of different things with that. So you want to make sure that your text boxes are correctly formatted as well. There are drawing tools in your PowerPoint that you can use to create your own schematics that you can use to enhance your poster. I will show you all those things in a second. You won't be penalized if you turn it in at least 72 hours before the presentation. No. Okay. So yeah, so the rest of it was if you were, especially if you were presenting in front of everyone, actually physically there. Since we will be presenting virtually from home, this becomes a little bit less important. However, you want to make sure that you do, like I said, present professionally and keep your presentation short and sweet. Okay. So I'm going to close this PowerPoint out and I'm going to actually go towards making a poster. For this, you want to go ahead and open up a blank presentation in your PowerPoint. Okay. So you will have a empty slide. You want to make sure it's a blank slide. I'm going to remove everything from here. Once you create your basic PowerPoint slide, you are then going to start to create. I'm talking about that in just a second. You are going to start to create the basic outline of your poster, okay? So to do that, we want to make sure first and foremost that it is the right size and it is the right dimensions and that you need for your paper, for your poster, right? So in our case, we were going to prepare a 24 by 36, so a 2 by 3 feet poster. That was our goal. We are going to change the size of this template, which is 8.5 by 11, to the size that we want. Now to show the grid lines, you're going to go under view and you will click on ruler. So you want to have the ruler showing, the grid lines showing, and the guides showing. These things are going to help you in aligning your different parts of the poster. Okay. Now you will go to design, and over here in the corner, you will find slide size. You're going to go to custom slide size, and you will make it the size you want. So in my case, I am going to put it to custom, and I'm going to say that I want a 24 by 36. So the height will be 24, the width will be 36. Now, having said that, you could do it either way. If you were going to be making a poster for the actual class, you could have made it in a portrait form or in a landscape form. However, since we are going to be presenting digitally through our screens, the portrait form is not going to look very good because it won't be able to enlarge the same way. So I would stick with the horizontal view. Okay, it doesn't matter what you put over here for now because we just need our poster template. This is just a blank screen for now. That's what you get. And now you see that you have a lot more grid lines in there because it allows you to then format it and align things very nicely on there. So that's our basic first step. We get all our basic first slide with the right dimensions on there and the grid lines showing. We will then go ahead, and if you want to format the background and you want to use one of these, for example, right? That's one thing you can do. You can use a different color. You can put one of the designs on there if you want, or you can just make your own color choices and you could do something yourself. Did not. So for the Mac, it doesn't do as many grids. You have to individually make the lines. I don't know. I forgot how you do it, but you like copy the grid line. You select it and copy and duplicate it multiple times and you get multiple ones. I don't know why. Otherwise, it only gives you one or two. I think like just four or something. However, as long as it's letting you show these guides, you're good. Because these guides are the ones that are movable. The grid lines are not. So it still helps you. I'll show you another way that you can align for the Mac users. But this is helpful. This grid line that moves is the best thing. The guides over here. Okay, so I'm actually going to change it to, I don't know, some other color. Let's do green. Yeah, that looks good. Why not? So you choose a background if you want to choose a background for your poster. I went ahead and chose something to show. Then you are, oh, no, Control C, no. Fine, I will do it again. Okay, I got my color again. Slightly darker version. Now you are going to go to home and you are going to start building your outline, or think of it as if you have a magazine page to do or a newspaper to do, you will do like your layout. So you're going to prepare your layout. And to do that, what you will be using are these boxes or other shapes that you may want to use to make those areas specified. The reason for that is that if I were to right now just start building on top of it, you will have all these little text boxes and images on this humongous slide. and it will be a lot more difficult for you to move them around and align them and make them the way you want. So by making out a layout, a plan for your poster, you minimize the amount of moving you have to do within different sections. So to start with, I will figure out where I will put my title, and it's going to be on the top. So I will make an area for that, right? And apparently I didn't select it. Okay. Size title. And I'm going to go ahead and make it a different color. That's going to be my title area. Then I will go ahead and mark out my areas for my introduction and methodology section. Now you don't want to have a lot of white empty space, right? Or a lot of wasted space. So you will notice that I'm gonna be leaving very little border on the sides. You want to maximize the use of your space. So when I'm making my boxes, I'm essentially making them to fill up most of that space. That's good. Okay, so I'm gonna do it the same way for now. I can give this one maybe an outline to help me see it. Now you can do Control D or Command D to duplicate your image, your box, and move it over to the other side. And you don't have to even move it. You could just select that and the figure above and say Arrange, Align, Right. And it's going to automatically align it to that edge over there. I will then duplicate this one again. And this time I'm going to move it to the middle. And I'm going to make it larger because this is for the central area. And I'm going to align all three of these to the top. I'll look at your text in a second. And I will align top. I know, Command D is amazing. Command D has a lot of useful things that it can do for you. I will show you some more in a minute. Okay, so you got a basic template going. Now, what you want to do at this point, you have your basic sections and you know where things are going to be. You are going to select all of it, right? So you can just kind of go like this and select all four things, four images. So they are selected together. You will go to Arrange and you will go Group. And now they will be grouped together. So if I accidentally move something, the whole thing moves. And it's easier to lock it back in place, just like so. You good? Perfect. That's what I wanted. So we have our basic poster template. Now let's go ahead and make some more little layout things. So we want to know the group option is not working for some reason. Is it there? Um, you may not have selected more than one thing. You have to select more than one thing for it to group. Are all four of your things selected, or two or three, however many you're doing? Huh, interesting. And yeah, you can do that as well. You can right click as well, and there should be a group option there. Try that. Noah always has interesting questions. Good. Also, sometimes if you have groups already made, it won't let you make another group until you ungroup it first and then regroup things together. Like if you have a group here and a group there, a couple of groups, and then you want to regroup them together, it wants you to ungroup first and then regroup all of them together as one. Okay, let's keeping it. Yeah, yes, I know we like interesting apparently. So good. Here, now we are going to make our subsections. So I'm going to select that again, and I'm going to go ahead and I'm going to make a little box here that's the same size as the one underneath, but now not as wide. I am going to give it a different color. Yeah, that looks like a nice heading color. There we go. There. Oh, maybe a bit of white on top. Up to you. That's your design. So that's going to be my introduction. I'm going to go ahead and write something in here to kind of get myself started on a thing. So I'm going to do 48. I'm going to do bold. I don't know. I'm going to do some interesting font. Which one should we do? Just for fun. Ooh, got so many cool ones. Make sure if you use a cool font or something, you use it consistently for all the headings or only the title, whatever. But don't make it too mishmash of things. Well, I'm not gonna go too crazy. So no, they don't have to be. None of it has to be in Times New Roman. However, the ones that look best for posters are Calibri and Arial and Times New Roman sometimes. Times New Roman definitely for the smaller text, but you don't have to keep it at that. You can be as creative as you want, but be consistent. So if you're using Times New Roman for your figure text, use it on all the figure text. If you're using a particular font of interest, let's say that I say that I want to use Algerian for my title. Well, that's okay for title. I wouldn't use it for everything, right? But then use it for my title alone. Um, or just for my headings, maybe for introduction, discussion, just for those words, something like that. It's a very formal font, though. Don't use it. You can if you want to. Anyway, going back to Arial Black. And it's going to be white. So let me write introduction. There we go. Oh, yeah, that's a good size. So I'm going to go ahead and use this to kind of orient myself on how big I want it. That seems like it's about right. It's not too small, not too big. And then I'm going to do Control D to duplicate it. And I'm going to put one down here. So I can have my aim and hypothesis, whatever I want to call it. So project aim. And then I have my methods. By formatting it once in the beginning, I don't have to do it again and again, right? So, methods. Okay, that looks good. And then you can always give them more or less space as needed, but this gives you a little bit of an outline on how much space it would be that you would need. No, so that would be that. Then I go and I select them, shift select, and then I do Control D, and I move it to the other side. Now the only thing that I that has to align is the top one, frankly, right? The rest of them obviously can move and be wherever they need to be. It's not a problem. But this one, I don't know what's going to go in here yet, so I'm just going to leave it like that. And then I will have my conclusions maybe at some point in life. I will have future directions or, you know, big world view, whatever. And then maybe I'll have my references. I want to make sure that all of these are aligned properly. I'm going to select all my grays. And I'm going to say arrange, align to the left. And I'll get those in place. Okay? So that's that. Now I have my figures. Let's do the central area. Now you want to make sure that the very first thing that you show up here, right in the central portion, is a showstopper, right? It's something to appeal to people, to bring people towards you, to look at your poster, whatever that's going to be. Could be images, could be just something interesting. Could be a schematic along with images, could be a graph with something of interest in there, but it should be something that pulls you towards it. Now for this one, it's still going to be bold, but my figure cap, usually my figure titles are a little bit wordy, so I'm going to make them smaller. Maybe I'll put it at 40, and it's not going to be capitalized. Caps lock is not going to be on for it. Let's say I just make up some things. I don't know. What can I write? Oh, no, that one's going to have your title in a second. Okay, so maybe that's going to be my first figure. And I'll show you how you are going to get your titles in a second. Um, and then something else. Who knows what that's going to be. But like I said, I am putting the titles of my slides Hello, guys. Hey, I'm back. Arr. Yeah, I know. It didn't give me any warning and just ran out of battery on me. So, let's start from where we were. Let me share my screen. My God, this is annoying. I have to see what's wrong with its setting to do that. Oh, come on, stop. Okay, so is everyone feeling good about their poster up till now? A lot of fun stuff still to do. You've been demoted? Why? What happened? Oh, oh, sorry. Well, thank you for taking over and helping my class along while I was missing. You can take over later again if you want. Sounds good. Well, my template disappeared, so some of it. I'll put some more things here. Great. So, remember, anytime you write in vitro or in vivo, it would be italicized. Just those two words. Seriously? Control D. There you go. Right seed extract. Needs to be Yep. Zoom University. Those two active central headings I can reset. Do that. Yep. So grape, seed extract, induces. I don't know. I'm gonna change it to activates gas phase 3. Or no, not activates. I don't know. My brain is not working. I have a typo. Where? Oh, reduces. Yeah, you're right. Thank you. Mm. Ooh, never mind. We'll do something else here. There you go. Okay, so let's go back to it and let's go to that top area, the one where we were going to put our title in. So I want to make sure that that title is going to be visible. It's going to be super cool, so people will come close to it, and it will make my uh, best, the overall conclusion very easy to find out. Um, so grapeseed extract, something about grapeseed extract has anti-cancer properties, or apoptosis, reduce. My brain is dead after my computer died. My brain is not working. Let's see. There you go. Something like that. So you're gonna write your title. Now, when you write the title, I put it as 72. I might even put it as 66, but it has to be large enough for people to see and know that it is the heading. Then you would go underneath and you would write the names of your partners. Mm. And these names, you are going to reduce the font sizes on those to maybe 48, 44, that's good. And then you would go down one more time and you would write Department of Biology. Department of Biology, University of North Florida. And then you will take this last line and make the font even smaller to maybe 36, 32, why not? And now you want to make sure that this stuff fits in the space that you have. So you may have to make it larger or smaller according to your needs. When you do that, you want to make sure it's not so uh, different that it goes on top of your next heading, but this works. There we go. The cell line in the title. You do need to specify that you're using some cancer cells or cancer, so in this case, I just wrote anti-cancer properties. So that is enough because it has to be, cancer cells. I would write in here in vitro. So they know that it's cell lines and not actual humans or mice or actual organisms to look at. As long as you have specified your cell line in your methods or somewhere else, you're good. You can if you want to, but it's more important if you are going to write something about the cells, It's more important that you mention that it's breast cancer cells or colon carcinoma, something like that. Okay. Then we will go to our wonderful internet and we will go to UNF logo online, and they have actually ones that they would like us to use. They have a image of that somewhere that they tell us. They go under marketing and publication. Custom logos, alumni, temporary logos. And you could copy one of these. When you do them, you have to make sure that you are not destroying their dimensions. That's what they ask. So I'm just gonna take one of those, copy that image. They have downloadable logos as well. If you search for it, you should be able to get them if you want. And then you are just going to paste it as a picture. Now you can change the size as long as you keep the dimensions, right? So you want to make sure to keep the dimensions the same and don't make it distorted. And you'll put that on one side. On the other side, you can put whatever you want. Okay. So now we've got some more information there, right? We have a basic system going. Now, before we write something in here, we also need to create some more spaces for those things. The reason is that if I were to try to put a text box in here, it's going to make this entire thing a text box, and we don't want that. So what we're going to do is we are going to take boxes. Again, we are very big on boxes. And you're going to put them in the space that you're going to be using for your introduction. And you can make it the exact same size. You can make it slightly smaller, whatever you want. I usually just kind of make it about the same size. You can make it into a different color if you want, or just leave it white, whatever it is that you would like to do with that. Okay. And then I will duplicate that, move it over here. So I have one on the other side as well. It's gonna be a little bit bigger. You don't need to do that in here yet because you are going to be putting in your images first, and then you will just need to make a little box for your text for the captions. I'm just gonna make a couple more though. I put one in the methods, even in the methods actually, unless you know that you're going to be doing text for methods, don't put anything in there yet. This one can go for conclusions. Now, one thing I didn't do, which I should have done, normally I do that, is that I go ahead and write out what font sizes and stuff I'm going to be using in these. So they are consistent the first time before I start to copy them. So I'm going to select all those 4 text boxes before I make more. You want to have at least size 24 font. You can put a team logo and put it on the other side. Yep. Why not? Go ahead. So you want to make it at least a size 24 font, probably higher for these actually. You'll probably do 36 or 32. For your conclusions and for your introduction, they're going to be in bullets. So go ahead and you can click on bullets in there to begin with. So when you start to write in here, it will do that. So I could say, MDA 468, whatever. And it should be able to make them into bullets. It's still very small. You may be able to write bigger than that. It depends on how much you need to write. You want to make sure the font color is what you would like it to be, right? This may have a schematic in there for all I know. Who knows? But if it's going to be text, it's going to be black in these. Or you can make it some other color. It doesn't matter what color you choose. So you could do that, right? I may actually move it like this because I may want to add some images. And again, these are ideas for you. It doesn't say that you have to do any of this. You could do whatever you want to do. Extract. I can go to images. See what I find. Oh, that's cute. Hmm. You can look through, you can find something of interest and copy it and paste it in part of your as part of your intro to jazz it up. Why not? And maybe I want to show that the chemical structure of the compound. So grapeseed compound, anti-cancer compound. And it will show you some examples. It may show you a pathway there. Oh, wow, how nice. It will give you a lot of different stuff that you can look through and find something interesting. And if you find something of interest, copy it and paste it in there. Just make sure to add the actual site that you're taking it from. For example, this looks like a very good image to use. And you want to use that, then that's what you're going to do. Obviously not because it's heightened, but you get my point. So that's what you would do with that, and then add information regarding those things. So here is a student's poster from my lab, and for her, she made, a couple of sections in hers where she talks about the da, 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 da. So in her case, since she's talking about breast cancer, she wanted to focus on that. So she has traditional breast cancer options. These are the things people do. They have toxicity. So she talks about that's the diagnosis. These are the treatment options. These are the toxicity issues. And saffron is something that can help with that. Um, and so she puts in that. Now for her, this is her work in progress. I had asked her to add the compounds. There were two different compounds in saffron underneath the image of saffron. So move the saffron up and then put the compounds, actual compound structures that are useful underneath it, and to put along with some information. So that would be that. Then for hypothesis, she has her, saffron was expected to have anti-cancer activity, tumor activity in MDA 468 breast carcinoma cells, since that was her focus. Then she has her methods. I'm going to enlarge her methods and show you an example of how you can use a schematic to do that. So in her case, she had the cell line, and then she did a lot of different things with it. Well, it wasn't just her alone, it was three different people, but they did a lot of different things. They did morphological analysis like you guys did, and she shows that by showing some slides and some staining, and then showing that you did microscopic analysis. She did some viability analyses with trypan blue, and with crystal violet. So for that, she chose to show the different time points they did, and a little bit of information on how it was done with the spectrophotometer in one case, with the microscopic analysis in the other. Then one of them did colony formation assays to look at the tumorigenicity. So she shows examples of a 6-well plate, and then she shows the actual method in very short form. So all she tells is the saffron treatment every 72 hours for how many days, and then you observed the results with microscope. Same thing here. So when you're doing the examples, methods like this, you are only showing the most important part of your experiment. So you're telling the name of the cell line, you're telling how long they were incubated with the drug and the drug dosage. In her case, she had multiple dosages. She made a table of it to insert later on. And that table went in here and also in this first figure. Um, you show how they were analyzed. So, if they were stained, you show that they were stained. If they were, if you did spectrophotometer, it shows that you did spectrophotometer, and then how you observed and collected the data. And so that's what in her case she's showing for each section. All this was looking at the invasion of the cells. All this was looking at tumorigenicity of the cells. So they had two different areas they were focusing on. And that's how you would do that portion. Now to do this, you would want to make a slide of methods. You do not want to make that slide directly in there, okay? You want to open you want to open your PowerPoint that you've been putting in your data in, right? Though, yeah, of course you don't find it anymore because I killed it. So I'm going to open a new one, but the PowerPoint that you just turned in that has all your data, you are going to basically take that same PowerPoint. I don't know if any one of these exist anymore in downloads. I've deleted all of them. You're going to take that same PowerPoint, and in a brand new slide, you can make a schematic to show your methods. So I will show you how you can do that. You will start off by making a little plate, right? Actually, that's not how you make a plate. Let's go to a cylinder. There you go. And you can make a plate of cells. You're gonna change its color. There you go. That looks empty enough. You write the name of the cell line in there. Maybe we want it to be visible, right? You write the name of the cell line to show which cell line you used. Then you want to tell that you did two different things with it, right? So you could put in or three different things with it, whatever it is that you want to show. So for example, I can show, I can use an arrow to make an arrow, make it black. And I can show that I did my experiment 2 times. So actually, I don't want to use this arrow. I'm going to use a different arrow. Really? That's not the type of arrow that I just said. That. Oh, maybe this. Yeah, that's better. Use this. Flip it. You can make a line to join it. Obviously, it didn't let you do that. And for this one, shape, shape, shape. Now, whenever you do, some design of your own and you want to make sure it remains the way you want it, you select both parts and group them. Then you can move them around, do whatever you want with it, and it will be happy. So I'm going to put it back up here. Okay. So now we did multiple things with our stuff. We did relative growth analysis. You did caspase activity data. You did morphology data. So you'll put your relative growth on one side, your morphology on one side, and your caspase on the other. And then I'm going to let you figure out how exactly you want to show that. But here are a couple of things to see. You can make your slides. You can make a 6-well plate for, you know, how you did with the protein. You can make a template for a 6-well plate. Shape outline. Let's make it white for now. Maybe. There you go. And then we'll make some circles in there. Now you only had 4 or 3 wells that you cared about, right? So you don't need to make a whole 6-well plate just because you had a 6-well plate. You can just make a 3-well plate. And you put those 3 wells side by side, right? Yes. And you make these arrange, and you go to align, and you align to top, and they are side by side. They look nice. And then my circles, I'm gonna change their color. I don't want that color. More fill colors. No, that's and then you go ahead and write the name of the treatments in each one, right? So this is your untreated. You want it bold. I'm gonna make it smaller right now so it shows. And then you wanna go in here. Seriously, I was next to each other, and you put, for example, mulberry low. In this way, okay. Mulberry high. It's being difficult with me today. It's so weird. Fine, be difficult. I will get you to work the way I want you to work. Okay, thank you for finally listening. We want to have minimal white space so it can be enlarged as much as we can once we are ready for it to be enlarged. You're going to collect it all together, group it, and then you can keep this to the side for now. You can control D, copy paste it. You're going to copy it, control C, and this time you're going to paste it as a picture. Once you do that, you can make it bigger, and it should maintain its resolution, and that will make it easy for you to do what you need to do with it. And then you can put it on this side or wherever you want to show your treatments. You can change them to different colors, by the way. You don't have to do it like this. You could do low as light pink, this one as dark pink, who knows. But all those changes would need to be made at this level before you put them in. So I could do it like this, and my untreated maybe gray because it's untreated. There you go. That actually looks better. So we're gonna move that. When you copy paste it as picture, you can no longer edit it. So you want to do all your editing on this PowerPoint slide to make what you need to make, and then move it to where you need to move it. And again, then you can, tell that you treated for 7 days, then extract protein, then spectrophotometer, and then you did the caspase activity. So you can write all those stuff here. And you would do that for all of these. You can take images from line for microscope, or use like the little images they have in their stock images to add a microscope or spectrophotometer in here. So that's how you would do your little flow chart. You can also just write it out as different steps instead of making any images in there. You don't need to put images. It just brings it a little bit more stuff to it. Once you are done with it and you are happy with the final product, I don't know what is going on here. You will take your image. So in her case, if you look in her slides, really? She has an image, that we had been working on back and forth. And in her case, she didn't listen to me either, and she had it in here. But you can, once you're happy with your final image, this is the editable image. You can copy it, group it all together, copy it, and then paste it as a picture into your poster. So I could bring it here, and I could do, paste as an image, and I get it, and then I can make it the size that I need for the space that I have. And you will have that as your image. In this case, this doesn't include my, obviously the drug treatments. In your case, you should have your the little table from your experimental drug treatments and dosage PowerPoint slide. You can just copy paste that image directly into this, or you can make an image with how many? You have 3 treatments, so 4, something like this. And you can add that to your table, right? With the dosages in there. So you could have something like that in here. I want to make sure that this is the size that you need, so it doesn't take over your space too much. Probably around 24 or 20. And then you can write your treatments in there and fill that out and use that for your treatments. So that would be that. Then for your images, you want to put your images in multi-panel figures. And again, I'm going to be showing you this as your example. And this has my points in there too, where she needed to make changes. So bear with me while I go to the area that you want. So again, what you would do is you would take those images that you have started to work on in your PowerPoint already. You're going to put them into new slides for yourself. You'll have 2 separate slides or 3 slides with all the data combined together. And for those slides, you will organize them the way you want to show them in your final image. So if we were to look over here, once you have all the data where you want it, you will copy it and paste it as an image into your final poster. For your image, you want to make sure that all your axes are at least size 14 font. All the words that you write in the caption are a minimum of size 24 font. And so here's an example of a multi-panel figure. She used images that were low magnification to overall show the quality of the cells. then showed higher magnification images to show close-ups of the cells to show the difference between the various treatments. Then she had her pie charts underneath to show percent normal versus percent nucleated, stalled apoptotic, like you guys did. And then she used that to specify which area was of interest. And then she looked specifically at percent death or percent apoptosis later on. So your first image may just be this area. You may just have your the three images. You can choose to show just the 40x or 400x, doesn't matter, or 1000x, whatever you want. You would show just one set of images and then a set of pie charts underneath. And that could be your first image. You would have a little space underneath. You would make a little, text box, and you would make it of the size you want. So you can add the caption to that figure underneath it. Okay. So this actually tells you exactly what I just said, that you want to add your figure caption underneath. You want only one set of legend for all the caption, for all the pie charts, instead of her where she had 4 different ones. You just want one set and then use the same color combination in all 3 of yours or 4 of yours. You want to have at least size 14 font in your panels on the edges, right, on the axes. So you want to make sure you change those before you copy paste them as images into your PowerPoint. And then you want to add your caption with at least size 24 font. So it can go down to 24. And then you would write down, figure 1, figure 1A shows cells at 400x magnification, reduced viability with increasing dosage. Figure 2 shows the way the cells are acting with the drug treatment, how they're changing. And then, this is looking at n equals 200 cells per slide or per condition, whatever that is. But you are using words minimally, so just the important information. Okay, and you can say n equals 3 fields per type. So for each figure, that's what you would do, and your actual image would be like again here she has A is some images, B is a graph with standard deviation and the significant difference. Questions about this? I don't know if anyone is there still. Oh yeah, you guys are there. Sounds good. Is that good? You guys are getting there? Okay, perfect. So that's pretty much, like what I was going to tell you about this. So unless I'm going to give you guys a few minutes to kind of go through and think about any questions that you're having, any problems that you're having. And then you're on your own to make your final poster, which will look something like this, but with your data and your stuff. And then once you have it done to this point, just like her, where I can make comments on each section, you can go ahead and turn that in as a draft. And I will go ahead and comment on it and make sure that you guys are on the same page, on the right track, and I'll point out any issues that you may be having. Yeah, so the way I would want you to split it, maybe one person can start working on the methodology and the introduction side of it. Whoever has the images in more complete form can start getting the figures together, right? And so one person can work on the schematics. Whoever is better at it, for example, can work on the methods. And you also have the schematic with your mechanism of action or the conclusion one, right? The pathway analysis and the conclusions, and they can start writing those things into this final poster. The other person get the images ready, send them to the first person, and they can copy paste them into the poster and put that in and add the captions and everything else. That's one way that you can split it up. Another way would be that, it just keeps those two PowerPoints separated so you don't accidentally start working on this big, large poster with your final images until they are ready to be put in there. You can also divide up the images if you want, figures if you want, and you can say, hey, I'm going to work on figure 1 and 2. Can you get figure 3 and the pathway done? And then as you can make a Google Doc poster file that you can both be adding things onto. Does that help? When you're uploading it onto my for the assignment turn-in, you will be doing this. You will be saving it first as a PowerPoint, then you will go back and you will save as, and you will do a save as a PDF. Otherwise, it will be too large to be uploaded onto Canvas appropriately. So save it as a PDF and then post the PDF of your poster into the assignments. Okay. Any other questions? Noah's computer is not doing any more interesting things. No, you're not writing a paper. This is it. The poster is it. You guys have last lab next week. We are presenting our posters and we are done. No, this was one of her source poster that she was working on. The poster is due before class, yes. But the poster draft is due this week, so I can give you comments so you can correct it as needed before your presentation. Thank you, Noah. Okay, guys, just ping me. We can do, I'll be doing a version of this presentation again any multiple times this week. You can join in if you still need more help. And, just ping me or email me and I can help you out through that as well if you get into trouble or get stuck somewhere. No problem, guys. Okay, have a good day. And I will talk with you later. Let's stop the share.